Good morning. I'm delighted to be in Arizona and speaking at the ARM MESA meeting. Avectas are tackling the key barriers to provision of affordable and scalable CAR-T therapies. In this presentation, I'll be demonstrating our closed, consumable-based CAR-T cell manufacturing platform and showing a workflow where we produce over 1 billion CAR-positive cells in 7 days at a modest MOI of 1. This is made possible by a unique consumable-based architecture, which we're showing for the first time. And you can see a glimpse of in the picture here. I'll unpack during the presentation. And though indicative of CAR-T cells, this platform has clearly got greater applicability. So as we know at this meeting, the science of CAR-T works, with likely 50,000 or more patients treated at this point. And yet globally, a tiny percentage of people who could benefit from this therapy do benefit from this therapy. And that is because of the challenges in manufacturing. To change this for the benefit of patients and for the benefit of the industry, frankly, sorting out manufacturing is critical. Current autologous products are manufactured through fragmented open processes, and these processes lead to high failure rates, labour intensity, and a situation where the facilities involved are unsustainable. Together, these barriers highlight why scale and affordability remain a central challenge for autologous cell therapies and set up the case for Solupor as a disruptive solution. So looking at the picture here, you can see our single-use consumable. To make this solution possible, we've packed significant value into the Solupor integrated consumable in the picture, enabling multiple workflow steps. For instance, activation, transfection, transduction, expansion, and harvest of cells. What's critical is that the cells stay within the device, they never leave it, and thereby we eliminate open processes and other things which would cause a risk to the final product. This consumable-based architecture also enables us to measure CO2, dissolved oxygen, pH, and temperature sensing. And our consumable architecture confers significant flexibility on developers from research use only to GMP, where the consumable can be used alone, it can be used in the conjunction with our core controller, and it can be used in a core and satellite process, which is the focus of my talk today. This consumable-based approach drives down platform complexity, batch failures, drives down labour input and facility costs. And here's what it looks like. So on the left, you can see our core controller, and on the right, you can see a series of satellite rockers. What's critical in this setup is that the consumable, which you can see centrally placed on the core controller, is capable of being decoupled from that core controller and placed onto the satellite rockers. This is unique in the industry, and it breaks the paradigm of having a single cassette to a single instrument, which is commonplace for integrated platforms in our business. So what we can effect by this architecture is the high-touch day one activities associated with CAR-T cell manufacture, for instance, transduction, activation of cells, and if it's a more complex product, editing. Those all happen at day one, and after those high-touch activities, the culture can be moved without opening the consumable onto the satellite rocker units. These units are highly rationalized, they're cost-effective, and they allow us to scale out. You can keep adding on, as you can see here, like Lego blocks here. You can keep adding on. The images in the the picture here are from about a year ago. We've continued to aggressively value engineer these products to drive down cost and increase scalability. So to unpack this a little bit more, we look at a workflow where we compare in the top corner a batch workflow which is made on a standard integrated platform, where the machine, the culture is held up for the full 7 days of the process on the device. And we contrast that with the Solupor integrated, in which we can get multiple batches started by commencing on day one and then handing off to the satellite units. So on the top panel, in a 7-day process, one could achieve about 40 batches a year on an integrated platform. So to scale this out, you end up with your proverbial ballroom or football field of integrated devices. Contrasting with that, the Solupor integrated, we can get 160 batches through, which represents a 300% input in uptick in throughput and a decrease in facility investment. Having 4 starts a week is conservative, and in many settings you could get significantly more than this, and the benefits would be commensurately larger. So we're unlocking parallel processing for autologous CAR-T manufacture. So now turning to some data. So in conjunction with our scientific advisory board, we developed a representative or model CAR-T process. It's a 7-day CAR-T process. And on the left-hand side here, you can see that we produce over multiple donors and multiple manufacturing runs, a remarkably consistent product where we're achieving greater than 2.5 billion cells, starting from a modest 150 million cells at an MOI of 1. Moving over to the right-hand panel, we can see that the transduction efficiency associated with these cells is between 40 and 50%. And this is over multiple donors. We've a vector copy number of 0.5, so it's a very safe product, and we yield 1.3 billion CAR-positive cells at the end of this streamlined process. So, moving on to the phenotype of these cells. So not just the number of cells that we're producing, but the phenotype of those cells. So on the left-hand side, we can see that we retain the stem-like phenotype, which is desirable in autologous products. So a high proportion of stem central memory cells and central memory cells, and a very low population of effector memory cells and effector cells. Over on the right-hand side, we can see, notwithstanding a growth of 17-fold expansion over the 7 days, that we have very low markers of exhaustion exhibited. So these cells are not exhausted, they're in great shape, notwithstanding that expansion. So then if we look at the cytotoxicity of these cells, once again at a modest MOI of 1, which confers the developer a terrific degrees of freedom in terms of how they want to develop their therapy. We see a potent product with between 40 and 50% cytotoxicity at the 5:1 effector to target ratio, and commensurate levels at other target to effector ratios. And I'll note for the eagle-eyed amongst you there, we're measuring cytotoxicity as a percentage of all cells, not just the CAR-positive cells. And on the right-hand side, we can see that these cells continue to produce interferon gamma as well, so the cells are in excellent shape. Finally, in terms of the data, I think uniquely we're showing that within the same device and platform, we can edit cells as well. And we're showing, starting from 19 million starting cells, that we can bring about a 38-fold expansion over a 10-day process. The 10-day process representing 3 days for the editing and 7 days, as described earlier. We get transfection and expansion within the same system. So switching gears for a moment to operational readiness. We've invested significantly in our regulatory pathway. We've filed a DMF with the FDA around the consumable and the controller device. We have significant digitalization developed in the system, where we've automated sampling and we've sensing of pH and dissolved oxygen within the system, as well as CO2 and temperature. We've a compliant batch record produced for each run, along with data integrity, as well as describing the run which has happened. This also orchestrates in that core and satellite architecture that I described. The chain of custody and chain of identity is preserved between the stations using that electronic batch record. We've established a supply chain in the US. It was one of the projects that I effected here from Boston, and have a network of qualified US suppliers. The devices are manufactured in San Antonio now. And the device is technology ready for transfer, whether at a research use only state or in a GMP state. So for the investors among you in the audience, just a brief word about Avectas, the company. We're approximately 30 employees based operationally largely in Ireland and headquartered in Ireland, with a number of US employees based in the Boston area. We're guided by an experienced management team from the pharmaceutical industry, and we have a world-class scientific advisory board who've steered the company scientifically and given rise to some of the great data that you've seen, certainly in the planning stage during this presentation. They include Bruce Levine, who's a luminary in the field, Mark Loudall, and Professor Everin Elisi. They're very excited about the data that we've produced principally, but more so about the architecture, the manner in which we've produced it as well. We've a broad IP estate comprising over 90 patents, both pending and granted, over 14 unique families. And we've raised in excess of $60 million to date. So in summary, I hope I've shown you today, first of all, that the key challenges for cell therapy at the moment is getting those therapies to patients. And the key barrier within that is scalability and affordability of therapeutics. We've set out a consumable-based approach that addresses the critical challenges and constraints in manufacture of CAR T therapies. We've also demonstrated flexibility, so starting with the early developer using the consumables essentially on their own, and then layering in the automation and the digitalization as one advances the therapy through the phases of approval. And lastly, that we are late stage, we're tech transfer ready. In fact, we'll be transferring the platform that I've shown today to a leading US CDMO within the next couple of weeks. So in terms of this meeting, Avectas is actively seeking partners who will work with us to commercialize and bring to the clinic CAR T therapies which are manufactured on our novel and unique platform. Thank you very much indeed.